just want to thank all of you for being here tonight, and a special thanks to Brian Fink for the invitation to share this presentation with you, and also to Father Steve for having the Lumen Veritatis series here at Resurrection. What a great gift it is for us to be able to come together every month and to learn about the good, the true, the beautiful. The inspiration for this talk, it came to me just as a free gift, really. I would have never chosen to necessarily do higher studies in biology or anything, but as a vowed consecrated religious, I have the vow of obedience, and through that obedience, I've received this as a gift. And part of what I'm gonna show you tonight is moments of wonder and awe that I experienced as I was going through my studies at the Catholic University of America. The title is The God Gene, Encountering Beauty in Molecular Biology. Somebody asked me the other night, what do you mean by the God gene? And I said, I don't really know. I just said my title was Encountering Beauty in Molecular Biology, and Brian Fink kind of added the God gene part, so we can ask him. So, like I said, or as Brian said, I graduated just in May from the Catholic University of America, and I owe a lot of gratitude to many people for that great accomplishment. And among that would be people that supported me at the Catholic University of America. Also those who supported me at St. Michael the Archangel last year, and my own community, the Dominican Sisters of Mary, and my community here at Lansing Catholic and Resurrection. During my first 2 years that I was here, I was writing my dissertation while first year teaching, and just trying to figure out the whole world of teaching and writing, and still finishing up experimental research. So I very much see God's providence in my accomplishments, and I'm grateful for that. I just want to share with you, because I know a number of people in this room and a number of people in the parish and in Lansing have been supporting me with their prayers. And this picture here, well, I lost my little pointer, but the one on the far left is a picture of my dissertation committee, which was taken after I passed my oral comps in just March of this year. As you heard, I was doing quite an amazing number of things last year. I was teaching 4 different preps, all new, founding a school, and preparing for a doctoral defense. And honestly, I flew out to Washington, D.C. from Kansas City for the month of March, or for the week of spring break. And my defense was the most peaceful experience I've ever had. And it's just the fruit of prayers. I know that many of the parishioners here were offering masses. I know the sisters were supporting me in prayer, not only here, but in Ann Arbor and across the nation, and even in Rome. So what a great privilege that is. I very much feel that this gift is something that will continue to grow with me. And maybe like some of you, it's something that I'm a weak person. I want to hide my gifts sometimes, and I know that the best way to use the gifts is to actually give them, and the more that I use them, the more that they'll flourish. So it's my first time to try to speak the language of molecular biology with the regular public, so I'll do my best to give you something you can understand. So one of the things that kind of inspired my talk was I just love the beauty of creation, and that's something that all of us can experience. I think any of us can think of beautiful nature, or the galaxies, or just even family life, and the beauty that we encounter. And through that beauty of encounter, it reflects something of God and His goodness. And in particular, there was a time when I was studying at the Catholic University where we went to the Air and Space Museum, and we went into the IMAX theater, and it was showing a presentation on the Hubble telescope. And I sat in that presentation, and I was just amazed. The Hubble was showing us things that had never been seen before. New galaxies being formed, stars being born, stars dying, the universe expanding. And looking at all of that glorious nature and creation that's outside of us and outside of this world was just remarkable. And what I want to do tonight is take the glories of God's creation, not from outside, but actually from within us ourselves. So not to go outward, but to go inward into the little basic unit of life, the cell, and explore the molecules that make life possible. One of my favorite, well, I wouldn't say necessarily favorite, but there's so many parts of Scripture that are inspiring. And in Job, it's always kind of interesting when God starts questioning Job and says, "Were you there when I founded created the heavens?" And so I pulled a quote from Job. "But ask the animals, and they will teach you, or the birds of the sky, and they will tell you. Or speak to the earth, and it will teach you. Or let the fish in the sea inform you. Which of all these does not know that the hand of the Lord has done this? In this hand is the life of every creature, and the breath of all mankind." What I would like to do is hopefully guide you tonight into seeing how God is present even in the little molecules that make life possible. So I'm not sure how familiar that is to each of you, but we'll see how that goes. So the cell is a basic unit of life, and just to give you an idea of how big, or actually how small that really is, there's this little YouTube video, which hopefully I can get to work. We'll see if it will. Let's see. Thank you so much. So this is showing you the size of different things. It's going kind of quick, but you can see the sperm and the egg. Red blood cells are 8 micrometers. The baker's yeast, which I use, the mitochondrion. Anyone know what that is? HIV, hepatitis, ribosome, antibody, hemoglobin, phospholipids, methionine, adenine. Those help make up part of DNA, and the carbon atom, which you need for life. So that. To scale. Yes. So that's to show it to scale. I'll show you that again because it goes kind of fast. And we got started without seeing. So you see the coffee bean. I'm a lover of coffee. And the sesame seed, the grain of salt, that's 0.5 micrometers, 500 micrometers, paramecium. Skin cells are 30 micrometers. Red blood cells. E. coli. So we're zooming pretty far in. Water molecule, it's picometers, pretty small. So that's just to kind of make the contrast before the grandeur of the galaxy and the smallness of the cell. And something that really struck me in my studies was in developmental biology. And something that we all share in common is that we all started at one point in time as the fusion of a sperm and an egg, and that formed one single cell. And that single cell, dare I try anything with this? That single cell, so your fertilized egg, goes through a series of rapid division. So you may or may not know that most of the DNA in our body is identical, and that's because we started as a single cell. So rapidly it divides from 1 to 2 to 4 to 8 to 16 to 32 to 64. And then it starts, so all the time it's communicating with itself, it's telling its neighboring cells what to do, where to go, how to form itself to get into the right position to make the tissues. So you've got the blastula phase, you've got early gastrulation where the cells have now multiplied enough that they're ready to start forming the body systems. And so it enfolds, and this is actually how your dietary system starts to form. And what's really cool is you form 3 different cell layers. And so you form the endoderm, the ectoderm, and the mesoderm. And through those 3 different layers, we eventually give rise to the endoderm gives us our gut, our liver, our lungs. The mesoderm gives us our skeleton, our muscles, and our kidney, our hearts, our blood. And the ectoderm gives us our skin and our nervous system. So that's just something to just be amazed about. Starting as a single cell, all those rapid divisions, all the things that could go wrong but don't, as you're going from a single cell to our bodies now, they are estimating, at least based on a paper that was published in 2015, we've got anywhere between 30 to 40 trillion cells that make us up. And we all started as a single cell. So that's just something kind of amazing to think about. But the cell is actually too big for what I want to focus on tonight. I want to go down into the molecular world. So there are 4 types of molecules that are essential for life, and these are carbohydrates, sugars, these provide energy, so that's why you like to eat carbohydrates, nucleic acids, which contain the genetic code. I'll spend a lot of time looking at our DNA tonight, and proteins, which give a lot of the structure and function to the cell, as well as lipids, which store energy. A cool thing about lipids is one of my first cellular biology classes, the professor was talking, he's like, it's just amazing that we are held together by little lipids, because our membrane is all made up of little lipids, and there's a bilayer, and that's what's protecting our insides from our outsides, and that's what's setting the gradient for our life. It allows a lot of things to happen, which give the conditions for life to happen. And something that I want to focus on in this slide, I'm not sure if you can see it very well. It's not the best quality photo. But with biology, you've got this idea of order and structure. So we have what's known as monomers for each of these molecules of life, meaning you just have one base unit. And when multiple of them come together, you get polymers. So you have many of those single things coming together to form a structure that has a specific function in the cell. So starches are made of sugars, basically. So when you have a collection of sugars, you've got a starch. So cellulose would be an example of that. With DNA, you have the basis of a nucleotide. So you have a nitrogen base, a sugar, and a phosphate group. And these come together and they build up the DNA that makes us, as well as other RNAs that help do various functions within the cell. You might have heard of DNA as chromosomes. So that's when your DNA is tightly wound together. You produce dense amounts of DNA that we called chromosomes. Proteins, these are kind of fun. So there are about 21 amino acids. And when proteins are being formed, you get a chain of those amino acids. So a protein, a polypeptide, is just a chain of amino acids together. Each of the amino acids has a specific property. It might have a charge or a size, little differences that set them apart. And after you have your primary strand of protein, it then folds up together in different ways, and sometimes it folds up together with other protein molecules or polypeptides to form proteins in our bodies. Proteins do amazing things. They give us all of the structures. They catalyze a lot of the reactions. They're just amazing. Most of our cell is made up of proteins that are, it's almost like little living molecules. They know exactly what to do. You've got some that are just carrying vesicles across the cell to very precise locations, and some are acting as little tags to let you in and out of the nucleus or to direct you to other parts of the cell, so specific organelles within the cell. And then lipids are again, chains of fatty acids. And so they're really great at storing energy. So I want to focus on DNA. And DNA is called deoxyribonucleic acid. I bet not everyone could say that one time, let alone 3 or 4 times fast. It's known for its hereditary material. So in all of our cells, we have DNA. And when our cells copy themselves, so when development was starting, you were making copies of your DNA. The DNA is basically the message that tells the cell what to do, like what to build, how to renew, how to repair, how to direct certain functions. And what's amazing about DNA is you've got 4 nitrogen bases, adenine, thymine, cytosine, and guanine. And these 4 bases create a sequence that encodes information, and that's what tells the cell what to do. So it contains the info for building and maintaining an organism. In our human genome, so the DNA in our cells, we have 3.1 billion bases. It's kind of a lot. And what's kind of amazing is all of us in here have DNA that is about 99.9% identical. So, so similar yet so different. And what's amazing with the structure, so you've got, if you remember, I was talking about, this is where I lose people, right? You've got sugars and phosphates and the bases that come together. So one of these is a monomer, and then when you have a bunch of them together, it becomes a polymer. And there's a beautiful structure here where we've found that the adenine always will pair with a thymine and the cytosine with a guanine. And that is kind of the basis for how the structure works. It's anti-parallel and it's got a helical structure to it. And what's neat is even Watson and Crick, these are the 2 scientists that came up with the structure of DNA. They were amazed at its beauty, basically. In April 25th, 1953, they published the double helical structure of DNA. They published it in Nature. And the structure was beautifully simple. That's kind of a key part of science, is often the more simple something is, the more accurate it seems to be. And they used it based on a couple of different modes of evidence. One of them was an X-ray crystal structure from Rosalind Franklin. And when they saw her image, it made sense. It was a double helix. And before that, there seemed to be some pretty crazy ideas of what the structure of the DNA would be. I love this little quote from Watson's memoir. It said, "The instant that I saw the picture, my mouth fell open and my heart began to race." Like the love of a scientist for their discoveries it's kind of amazing. And this is showing them with their model of DNA. So it's got the double helical structure. Watson and Crick and Wilkins received the Nobel Prize in 1962 for this discovery. What was kind of neat is as I was searching the web, I saw that Francis Collins compared DNA to a rose window once. And so since I was talking about beauty and DNA and molecular biology, I thought, well, that's kind of interesting. If you don't know, Francis Collins was the director for the Human Genome Project and just did tremendous work for sequencing the first human genome. So you can judge for yourself how similar the rose window is to a structure of DNA. It's kind of amazing to see molecules sometimes in the forms that they're captured in. What I love about Watson and Crick's discovery is that the DNA structure itself had a meaning of a function already hidden within it. And so it's in their Nature publication, it said, "It has not escaped our notice that the specific pairing we have postulated suggests a possible copying mechanism for the genetic material." So if you remember, as you go from a single cell to another cell, you're copying all of that genetic material, all the 3.1 billion bases that are present in a nucleus packaged very tightly. And so what's amazing here is within the design, you've got this double helix.
I have an easier pointer for you,
Oh.
so that you don't have to work the mouse.
Thank you. Awesome. So what's amazing is that the double helix nature, it's all held together by hydrogen bonds. And so hydrogen bonds are together, they're really strong, but individually they're weaker than the covalent bonds that are actually holding these nucleotides together. And so they're able to pull apart, and then you've got a way for other nucleotides to be added in. And so one of the amazing things about proteins is there are enzymes. So one of them is a DNA polymerase. So this is a protein, and its specific function is to take free nucleotides that are present in the cell and add them one at a time. So it'll match it up with the A's and the T's, the adenines and the thymines, and the C's and the G's, the cytosines and the guanines. And it'll make an exact copy of the DNA that is being replicated. So this is known as DNA replication. There are many, many, many proteins that are working with this type of a process, that are pulling apart the DNA strands, that are keeping the DNA from becoming supercoiled. It's kind of breaking and releasing and untwisting it so that it can actually be copied. And at the end, when it's all finished being replicated, you end up with two new strands of DNA. One of them old, the one that existed beforehand and was used as a template, and then the brand new one that was brought in to form the new strand of DNA. So it's kind of just amazing to think about. And that's happening in all of our cells. So 30 to 40 trillion cells, whenever they need to be duplicated, they go through this type of a process. And sometimes there are mistakes that happen, but even within this machinery, within these proteins, there are mechanisms that will help repair any type of mutation that might be put in. So if you get the wrong base put in, it could be passed on to future cells. And sometimes that could cause terrible effects. Sometimes it doesn't harm the cell at all. And sometimes it might provide some sort of advantage. But that's all present just within the nucleus of the cell where this is happening. So I want to talk about dogma a little bit. This is a group where I think we can talk about dogma, but it's not the typical dogma that you might hear of. This is the central dogma, and so I've kind of focused on it a little bit already. There's the DNA replication, so that's where DNA, did I break it? DNA goes back to DNA, but there's also how do you form protein? So this is kind of an amazing part of molecular biology. So DNA has the ability to pull apart, and similar to replication, it can produce an RNA transcript. So only one part of the DNA is actually going to be used as a template. So you're not replicating the DNA, but you're creating a new type of molecule. And this is a messenger RNA molecule. It's just a single strand, and just like the DNA that it's coming from, it's carrying specific information, a specific code, which will tell the cell what protein to produce. And so just like you saw with the replication, there's amazing protein machinery that comes together and will take the messenger RNA, and it will take transfer RNAs, and it will match up the bases. And as those bases match up perfectly, every 3 messenger RNA bases will code for 1 amino acid. And so little by little, you get an amino acid, another amino acid, another amino acid. And so you build this protein over time. And that protein is going to have a specific function within the cell. So what I want to try to do is take that basic idea of how DNA can be replicated in the cell, how the DNA can be used to encode a protein, and show how it can be applied in science. This actually isn't part of the Sup 5 work that I did for my dissertation work, but it's a project that I did in my first semester that I was in the lab. And it's just kind of a little bit more simple, and at that point in time, everything was so new, it really struck me, some of the procedures that I learned and I did. So they blew my mind. I'm hoping that they can blow your mind as well. It dealt with the cystic fibrosis protein. And so when you have mutations in the cystic fibrosis transmembrane conductance regulator, I'll call it CFTR, that's a lot easier, into that gene, it results in cystic fibrosis. And this is the most common lethal inherited disease among Caucasians. There's about 1 case per 3,200 individuals. This disease is autosomal recessive, meaning that you need to get a copy from both mom and dad to have the phenotype. And what it does when you have a mutation in this gene is it causes a disruption of the salt and fluid balance, affecting absorption and secretion. So if you look down here, this is that awesome lipid membrane that separates inside and outside. So the barrier of the cell here, and here you've got chlorine molecules, and here's CFTR. And CFTR is that protein, and its function is to allow chlorine to go into and out of the cell. And what happens when it's defective is these molecules are unable to leave, and then the patients with cystic fibrosis end up getting thick, sticky mucus in their lungs and other exocrine tissues, and often they will die of some sort of bacterial infection. The median age of survival is 37, and it leads to lung disease and chronic infections. So that's some background. If we look at the CFTR gene, so a gene is a portion of the DNA that holds the information that encodes the protein. So if you look on the chromosome, it's founded on chromosome 7, and the gene was identified in 1989. It's located on the long arm. Our chromosomes have long arms and short arms, P's and Q's. It encodes the chlorine channel, like I said before. And there are more than 1,500 different mutations identified in the CFTR gene. But the most common mutation is an in-frame deletion that leads to the absence of a phenylalanine residue at 508. So that's fancy verbiage, meaning that chain of amino acids, when you get to 508, that amino acid disappears. And so when that disappears, it produces that protein that has a defect in the chlorine channel. So the CFTR gene encodes the CFTR protein. And it's a huge one. It weighs in at 480 residues. It has 2 transmembrane domains. So if this is the plasma membrane, so the membrane on the outside of the cell, the transmembrane domains are going through that lipid layer. And these are the parts that form the channel. So they close and they open to allow the chlorine to come into and out of the cell. It also has a series of domains here. There's nucleotide binding domains. The sisters were just reading my dissertation last night, and they're like, it's all these letters and numbers. It's like its own language. It is. Yeah, so I'll try to explain it. And so these domains here, they will bind ATP, which is the energy source for the protein. When it's able to take the ATP, it gets energy to open up the channel and allow the chlorine to exit. And there's also a regulatory domain that's present here, so it functions with the nucleotide binding domains. Hopefully that makes some sort of sense. So the function of the CFTR protein is to allow chlorine to enter and exit the cell. And that deletion of phenylalanine would be in this domain here, the nucleotide binding domain. So that's where it's lacking that one amino acid, which is amazing. A mutation in one nucleotide of DNA can cause a disease. Doesn't that make you so grateful for all the things that really go right? I mean, it's phenomenal. I love this slide. Again, not knowing your background knowledge, we're looking at the inside of the cell. There are many organelles in the cell, which are like organs to the body. So they're regions that have specific functions. And so your nucleus is located in here. Your nucleus is what holds your DNA. And as your proteins are being produced, they go through a series of organelles. They start in the endoplasmic reticulum. CFTR is actually packaged into the cell membrane, so it actually gets inserted into the ER membrane as it's being produced. Again, amino acid at a time. And as it goes through, I love it, it will eventually bud off, so these little vesicles will form. So these little dots are showing CFTR protein being produced. And it'll actually go across the cell to another organelle, the Golgi apparatus. And the Golgi is amazing also. I might be a little bit biased. And as they go through the Golgi, they mature. They might get extra chemicals put onto them, extra tags put on. There's little sequences on some of them that tell them where to go. And they just kind of go through this maturation process. And then ideally, they'll then be sent to the plasma membrane. So that's where they're going to function the best. That's where they need to be to allow the chlorine to exit the cell. And what we find with the delta F508, so that defective form of CFTR, it actually just ends up being degraded by the dump of the cell, the proteasome. So it will just take that protein and it'll chew it up, and then it'll recycle those amino acids so that they could be used in a better place. So it's just kind of amazing, all that's happening. So I kind of inherited this little project my first month in the lab, and the idea was, okay, in my lab we study the yeast. Yeast are about 26% similar in their genes to humans, so there are some genes that are pretty similar to genes that we have. And PDR5 is the name of the gene that I studied most in depth. It is in the same family as CFTR. It is a transport protein. It is found in the plasma membrane of the yeast cell, and it will pump drugs and toxins and thousands of different types of toxic things to the yeast out of the yeast cell. So it's kind of a unique type of protein. Some are extremely specific to one type of substrate, one type of molecule, but PDR5 will take a lot of different things. So it's kind of unique. CFTR and PDR5 are both found in the ABC transporter family, and ABC transporters are found in all types of organisms. So it's a very common type of family of protein, and the idea is we can study the ABC transport proteins in yeast and gain understanding for the ABC transporter proteins that we have in our bodies. PDR5 is kind of unique though. It's very different from almost any other other ABC transporter. So this is a little map that shows relationships between genes. The closer that the lines are to each other, the more closely related those genes are to each other. And you've got a couple of different species going on here as well. So I'm going to point out over here, you've got SCPDR5. So that's the Saccharomyces cerevisiae. That's the fancy name for the yeast. And that's PDR5, the protein that I study. And if we come here, let's see here. I lost it. Oh, there's the mouse CFTR gene. There's the human CFTR gene. So you can see how similar these genes are to each other in terms of their sequence, and how far apart they are. So they're in a similar family, but they're pretty far apart. So the question that we were asking is, if I delete a similar residue in PDR5, will it affect its ability to go to the plasma membrane, like we saw with CFTR? And actually what we found with CFTR, not me personally, but the scientists in general, is that it's a result of misfolding. So just deleting that one amino acid prevents it from being able to fold properly, and then it is not going to make it to the membrane. So there's a series of steps that I was going to take the first time in the lab. The first one is called site-directed mutagenesis, and that was one that just blew my mind, that I really wanted to share with you. But I also wanted to kind of share a general simple procedure with you in case that it also kind of was amazing just to hear what we can actually do in molecular biology. So let's go to the next slide. So this is a plasmid, the circular piece here. So a plasmid is a fancy name for a circular piece of DNA. And with this site-directed mutagenesis, I could design a little piece of DNA, we call it a primer, and in that design, I delete the nucleotide that I want to delete the amino acid in the end. So I can create specific deletions in the DNA wherever I want it. It's just in how I decide to design it. And then, so that's what these here are showing, is those little primers, those little strips of DNA that I would design. And on the plasmid, I would have my PDR5 gene, and that would allow the primer to find that gene so that it could cause the deletion to happen. So the way that this process works is basically you take your DNA, you take your primer, and you take the polymerase, so that protein that adds the nucleotides one at a time, and you take your nucleotides and you stick enough in there so that you can create a new DNA molecule. And you basically vary temperature. And so when you put the temperature at a very high temperature, you melt the DNA, so the strands will actually separate out, which allows the polymerase to bind onto the DNA, where it can add those nucleotides. And you cool it to the right temperature so it can do those additions, and it goes through tons and tons of cycles, so that by the time you finish doing this reaction, you've got billions of molecules of DNA that you've mutated, as well as some of the original DNA that was started in that little reaction. So this is not in the cell, this is all in a test tube, which is kind of amazing. Like I said, it's like the proteins are little living persons able to do this type of thing. They're so specific to what they're doing. And then the system just is designed as well. By the end, you can add another little enzyme that's going to digest all of the proteins that don't have the mutation, so that you don't have a mistake. And then what you do is you can take that little plasmid, and we actually use E. coli. So we stick the plasmid into E. coli so that we can grow lots of it. And then we can make sure that the mutation is actually present. So we can sequence it and make sure, oh, yep, it's deleted, I want to use this one. And then we can take it and we can stick it into our yeast. And once we put it into the yeast cell, we can then study the phenotype. So one of the ways that I could see if this mutation was affecting my yeast is it's a drug transporter. So the black line is showing what the yeast cell typically is able to do when it has this amount of drug added into a test tube. So this is just a normal, healthy yeast cell. The R1 is a yeast cell that has PDR5, my protein totally deleted. So if you don't have PDR5, you have instant cell death. Where's my little pointer? As soon as you challenge it with any bit of drug. And this R263 was that deletion that I made. And I could see as soon as the deletion is present, even though PDR5 is still present, the deletion of that one amino acid was enough to kill the function of PDR5. And I actually don't remember because that was long enough. I can't remember. I also tested if it made it to the plasma membrane. And I don't think it did, but I can't quite remember that part. And then you can also test other parts of it as well. So hopefully you were somewhat amazed. I don't know. Hopefully you encountered something beautiful. I just think there's so much in that little world of the cell that amazes me. And hopefully you could take in some of that. Like I said, I mean, the number of things that could go wrong at any moment and the number of things that go well helps me to realize that, gosh, I mean, there has to be a God, right? I mean, it's just kind of amazing all that goes right so much of the time. And early on in my studies, I was just amazed by the cell, and this scripture passage kind of struck me. As a body is one, though it has many parts, and all the parts of the body, though many are one body, so also Christ. Normally, I think of that passage in terms of like the body parts, you know, like the arm, or consecrated religious are the heart, right? But but I was thinking, gosh, and I actually asked Google, like how many people had ever lived, because 30 to 40 trillion cells, like what if we're all little cells of Christ's body? And you think about when you're growing in virtue, you're growing in all the virtues, and when you're growing in holiness, you're helping people around you to grow in holiness. But the same is true when you're not choosing the good, when you're not choosing to be a believer and to be faithful to all that you've been given. So you think about a cancerous cell. It's very selfish. It takes all the nutrients from its neighbors. It causes destruction. It grows out of control. It starts crowding people. Just, I don't know, it's amazing. But also there's treatments for cancer and for other diseases too. So even though you might be cancerous or you might have a defect, there's still possibilities, and maybe it's through your neighboring cells that send you the right signals that can help you to bring more health to Christ's body. I don't know, just a little meditation there. But hopefully, I see eyes open, so that's a good sign. But that is the conclusion of my presentation. If you have any questions, I'd be happy to try to entertain them. Father Steve promised that there would be questions and answers. So if I don't have an answer, maybe some of you do. We'll take this young man in the front.
Young man, I love Dr. Stephen Patrick Joy. Congratulations. So, you took me back 50 years when I studied genetics as an undergraduate Bible major 50 years ago, at least. A lot of what you talked about wasn't invented then. I mean, Watson and Crick had been around, it was pretty cool. But we didn't know all that stuff. Nowadays, you can get your whole DNA sequenced. Our daughter had it done. It's a 500-page report. She knows every gene that could cause some problem in her.
All you have to do is spit in a tube.
Yeah, right. She actually had it done by Craig Venter, who ran the Human Genome Project. The point I am is worried. I'm truly frightened. I'm not frightened by much, but I'm truly, truly frightened by where this is all going. You know, Can I order my next grandchild to be 6 feet tall, green eyes, blonde hair, beautiful, 150 IQ? With CRISPR, I probably could. They can edit any problems out of the genome. It's a scary world where we're going. Help me understand it better. Tell me, what's the future holding?
You need to pray. You did, and I'm so grateful for that. Yeah, it's amazing what you can do. Oh, CRISPR-Cas is a little bacterial system, so it's basically when there's foreign invaders, it can take that DNA and clip it and stick it into its genome, so it's like a immune response. So basically any foreign invaders that come back that have already been there can then be identified and wiped out. But we've now taken it and you can edit any gene basically in the mammalian genome by sticking certain sequences in specific spots, kind of like I explained with PCR, but a slightly different system. So it's really, and it sounds like it's gotten more and more precise too since I've studied. So there's a lot you can do. And as a biologist, I mean, I feel confident, like I could do anything almost that you think of, like with enough thought and with the tools and with the money, there's really a lot you can do. But what's amazing is we're always manipulating what the Lord has given us. Like we still can't create any of the life without something else that's giving us the life. And I read an article by a gentleman in organic chemistry. He synthesizes a lot of different molecules, and he was very frank. I mean, if you look at any part of the cell, if you look at the lipids, the lipid bilayer alone is incredible. The different combinations, the compositions, the proteins, the modifications on the proteins that actually go into building just a simple cell wall can't really be duplicated like our bodies do it so well and other organisms do it so well. So there's a lot that we can do, but there's still a lot that we don't know. And I think that's kind of the beauty and the mystery is we can never exhaust God's creation. Like he is so great. With the technology though, really you want it in the hands of the right people. Even with giving your human genome to your medical doctor, great things can happen. You can detect breast cancer before you even get it. You can do just so much so quickly, but there's again so much that you don't understand, that we as scientists don't understand. And so it's how much do you trust the person that is giving you those results? I don't know, it's kind of, I think prayer is a good option, and people do what they can. Like people always try to go beyond. And that's the importance of us witnessing to the faith, I think, of why you wouldn't cross some of those lines. But I'm not sure that answers your question at all.
It's 50 years from now, what it's going to be like. 50 years. I mean, I'm just saying that technology is advancing.
In my lifetime, I mean, it's been incredible, and I'm young.
There, but I'll be gone.
Yeah.
But it's remarkable how it's changed.
It really is.
I mean, from 50 years ago, when I studied genetics.
Yeah.
And by the way, I liked your quote from Job. I'm into trees, which are not subcellular.
That's true.
Anyway. But in any case, I mean, 50 years from now, I mean, it will be remarkable. I mean, 50 years ago, we didn't have PCR, you know, the Xerox machine for DNA.
Right.
Copy it and make as much as you want.
Right.
Run it through a PCR, polymerase chain reaction. And now, but from 50 years from now, what will it be like? scared. I'm truly scared. I mean, seeing this kind of stuff, this is so complicated, you can't help but believe that, you know, God invented this.
Oh, yeah.
You know, this isn't just random.
Sister, on that point, I'm curious. So one of the great hurdles right now with students, perhaps at Lansing Catholic, other places, is science is an impediment to belief in God, right? I mean, this is what you hear. So what you've laid out, 30, 40 trillion whatevers, right? PCRs, ABCs.
CFGs. No.
Right, okay. As you're talking about this, talk a little bit about how your students respond as they see the mystery that is becoming unlocked. How do they respond to that in terms of faith?
I think you get a varied response just based on the disposition of the student. I actually haven't had the privilege of teaching biology too much. It's mostly been chemistry. But when I have taught biology, like I loved it, AP biology last year, the students, you could see their minds were blown, and the few that were in the class really weren't strong in their faith either. So it was just kind of like, wow, this is amazing. Like, I've never heard it before, which is always my goal as a teacher, is to get them to see it at that level. But what I've also noticed is, especially with like stem cell research, there's so much that's not known out there with just the different types of stem cells, that adult stem cells are actually ethical to harvest, that they've actually got a number of proven therapies that have worked, whereas embryonic stem cells, we really can't control them, and we've not gotten any, as far as the last time I checked, results from any type of therapy or treatment that's tried to be created from them. So that's kind of a side note. Yes.
Sorry, I almost want to step away from the biology for just a second, if I can. Not my strong suit, or physics for me. But could you comment a little bit on the fact that you can know this? Because it seems to me, I'm listening to you talk, and I will readily admit, like, Father, I'm not sure I understood all of it. But it seems to me it's just as amazing, not just that it exists, but that God gives us this capacity that the I can learn this part of it. Or, well, you can. But there's a wonder there, too.
Yeah.
In the each of us can see a part of creation and appreciate a part. I mean, not obviously the entirety of it, but just, and the more we bear down and pay attention, the more it seems to open up.
Mm-hmm.
I'm just curious of your thoughts on the experience as much as anything else.
The experience has just really been mind-blowing. I mean, it's kind of interesting. Well, this is kind of changing the subject, so it's not answering your question. Maybe I shouldn't go there. But I've been in situations in the classroom as a college student where I'm sitting in my biology class waiting for it to start, and a person will come in and see me, look puzzled and leave, and then come back. And then they'll be like, is this biology class? Because I'm a Catholic nun in the biology science classroom. So it's it's fun to see those types of responses. It's odd that science is pitted against theology. It's odd that that has been seemingly to be the dominant thought overall. And I've been blessed. Recently, this summer, I was able to attend a conference with the Society of Catholic Scientists in Washington, D.C. I just happened to be there when they were there. And it was so beautiful and inspiring to be there with, I forget how many of us were there, but all these scientists and Catholic and wanting to grow in our faith and wanting to share that faith. The society was founded just, I believe, 2 years ago, and I think it's grown to over 700 members now. So we've got movements like that happening, which are just really inspiring. And then there's people like Father Robert Spitzer, the Jesuit who has a Magi Center, who brings a lot of the faith and reason into the public sphere. So I love that you can actually find more of the Catholic witness in the world. I think there still needs to be more of that. My experiences as a molecular biologist, it's very, it's very emotional actually. You really get tied into what you're doing. You have a lot more failures than you have successes. You have this idea of what you're trying to do, and there's a billion things that can go wrong, including yourself and your skills, but also you're working with life, which is also has variables that you can't necessarily control. So it's fun because it keeps you engaged and you're always wondering what's the result going to be. You have your highs and then you'll keep testing that thing because you want to know, is this really true? Is this really true? Is this really true? And what's amazing is as you're seeking that truth and as you're seeing the results, you're getting kind of a reflection of God in that. It's always kind of heartbreaking though when you finally disprove yourself. Yes.
As a teacher, have you seen it's a YouTube video on all the proteins that are made in the cell that are needed for a white cell to go outside from within the vessel, the blood vessel, to outside the blood vessel to go to the site of say, like
infection.
the skin.
Yeah. No, I haven't.
It's beautiful. It's a beautiful thing. I think it's I can't remember the name of it. It's been several years since I've seen it. It's so beautiful, and it goes along with what you've been talking about.
Yeah, there's also a beautiful video. It's got different lengths. I think one of them's like 5 minutes long or whatever, where it's the life of the cell. I absolutely love that one. I usually show it to my students if I'm teaching biology because you see these proteins alive, essentially. I love it. There's the little motor proteins that are carrying the vesicles across these microtubules, and they're just, trudging along, and you just think, gosh, it looks like so lifelike. It's amazing.
Sister, you mentioned, or some of us mentioned earlier about DNA, and does it make sense to you that with lab results of a little bit of saliva, that they can try to figure out that for the last 200 years your great-grandparents were from this country, but back in the 1600s, a couple of them were from Germany or France?
Yes, because there's cells, and there's single cell sequencing. You can get the genomic material from a single cell, any single cell, with great accuracy now. It's mind-blowing. Yeah.
So I just wondered how scientists are accounting for, are scientists accounting, let me rephrase. Is there greater and greater mutation on the cellular level in the human being than there used to be? Or are we just more aware of it now because of scientific development? And if it is the case that there is greater mutation causing a variety of diseases and illnesses, do scientists pin anything particular, like the food we eat or the environment, etc., as the cause?
Way to throw an easy one at me. Oh gosh, like the sequencing over time, that's kind of a remarkable thing. That's how you can get a lot of the ancestry, I would imagine. I haven't actually read a lot of the papers or followed a lot of the ancestry type stuff, but I would imagine you could actually see the evolution of specific genes through time. I haven't looked much into that for the human species. I should have written down your question. I'm forgetting all the
I'm
parts.
just curious, sister, if we're seeing greater and greater illness on a molecular biological level, is it that we're recognizing it because of scientific development, or is it really the case that it's happening more? And if it is the case that it's happening more, do scientists point to a cause like the food we eat or the environment or other factors?
Scientists are definitely looking into those factors to try to tease it out. You could probably find research that says, yes, it is, no, it isn't. So I don't think I'm going to have a strong answer for you. But we are also living longer just with modern technology, which as our chromosomes are duplicated, their telomeres, the ends of those chromosomes get shorter, which just kind of reflects the age of our own bodies and how long they can be repeated or replicated. So the longer you're alive, the more mutations you do pick up, which is why as you get older, you do tend to have more of the cancer, that sort of thing. But there's other things that can cause that as well. Yeah. Does that help?
Yeah, absolutely. Absolutely. Yeah. All right, well, let's thank you, sister, one more time. Thank you very much. Let's go.